Hello everyone. So let me share my screen. Okay. Okay. So good evening, guys. Today is the 11th live session. And as you know that in these live sessions, we basically discuss some sample questions. In these live sessions, we basically discuss some sample questions from last year's assignments. And we not only discuss the questions, but we also try to discuss the concepts behind those questions so that it's easier for us to solve different types of other questions, right? Right. So, and if we have any issues, we can discuss the questions again. And we try to keep these sessions as interactive as possible so that if you are facing any difficulties, we can discuss it out. So if you are not able to understand anything, you can just let me know by raising your hand or just unmuting your mic and letting me know. If I know the answer, I'll tell you right now. If I don't, then we'll discuss sometime later in the next class, right? So moving on to the first question for today. The molar ratio of vector to insert for a ligation reaction is ideally option A 1 is to 1, option B 1 is to 10, option C 1 is to 3, option D 1 is to 5. Which one do you think is the correct answer? I'll wait for 5 minutes and you can try to solve this and then we'll discuss. Okay, so we'll move ahead with the discussing these questions. This question. So basically, the answer will be 1:3. So there can be various ratios of vector to insert, depends on what the purpose is and what your starting material is, right? So depending on that, basically we choose the vector to insert ratio. Now let's discuss which one is the most prominent and when are the instances when we can choose or play around with this ratio, right? So first, if we have to talk about this vector insert ratio, we need to talk about start from the basics. So what is cloning? So animal cloning is an assisted reproductive technology similar to artificial insemination and embryo transfer and in vitro fertilization. So the current technique used for animal cloning is somatic cell nuclear transfer, SCNT. So there is basically no published use guidelines for unique requirements regarding the care and use of animal clones in research. So the care and use of animal clones in research does not differ from care provided for conventional animals to assure good animal welfare and animal well-being. In addition, because the progeny of animal clones are not clones, clearly progeny do not require special consideration, right? So what is cloning? Basically reproducing a genetically identical offspring. Now, the possibility of human cloning raised when Scottish scientists at Roslin Institute created the much celebrated sheep dolly. You must have read about dolly. If not, you can go through this nature paper that is mentioned over here. It's fascinating. So this aroused worldwide interest and concern because of the scientific and ethical implications. So obviously genetic engineering and cloning are a big boost to the genetic engineering community because we can do a lot of things based on the scientific manner, but also there's some ethics. Till at what stage do we stop? What are the organisms that we can manipulate? So there are various ethical implications as well. So this feat cited by the Science magazine as the breakthrough of 1997, it also generated uncertainty over the meaning of cloning, an umbrella term traditionally used by scientists to describe different processes for duplicating biological material. So what are the different types of cloning? There are different types of cloning, usually recombinant DNA technology or DNA cloning, reproductive cloning and therapeutic cloning. Now, the terms recombinant DNA technology or DNA cloning, molecular cloning, and gene cloning, these all refer to the same process, the transfer of a DNA fragment of interest from one organism to self-replicating genetic elements such as a bacterial plasmid. Now, the DNA of interest can then be propagated in a foreign host cell, and this technology has been around since the 1970s, and it has become a common practice in molecular biology labs today. Now, scientists studying a particular gene often use bacterial plasmids to generate multiple copies of the same gene. Now, plasmids are self-replicating extra chromosomal circular DNA molecules distinct from the normal bacterial genome. Now, plasmids and other types of cloning vectors were used by Human Genome Project researchers to copy genes and other pieces of chromosomes to generate enough identical material for further study. Now, what do you mean by recombinant and all of that? So if to clone a gene, a DNA fragment containing the gene of interest is isolated from chromosomal DNA using restriction enzymes and then united with the plasmid that has been cut with the same restriction enzymes. Now when the fragment of chromosomal DNA is joined with its cloning vector in the lab, it is called a recombinant DNA molecule. Now following introduction into suitable host cells, the recombinant DNA can then be reproduced along with the host cell DNA. Now, plasmids can carry up to 20,000 base pairs of foreign DNA. And besides bacterial plasmids, some other cloning vectors include viruses, bacterial artificial chromosomes, and yeast artificial chromosomes, which we call BACs or YACs. Now, cosmids are artificially constructed cloning vectors that can carry up to 45 kilobases of foreign DNA and can be packaged in lambda fast particles for infection into Escherichia coli or E. coli cells. Now, BACs utilize the naturally occurring F factor plasmid found in E. coli to carry 100 to 300 kilobase pairs DNA inserts. Now, bacteria are most often used as the host cells for recombinant DNA molecules, but yeast and mammalian cells are also used. Now, Dolly, the first mammal to be cloned from adult DNA, was put down by lethal injection in 2003. Now, reproductive cloning is a technology used to generate an animal that has the same nuclear DNA as another currently or previously existing animal. Now, Dolly was created by reproductive cloning technology. By a process called somatic cell nuclear transfer, scientists transfer genetic material from the nucleus of a donor adult cell to an egg whose nucleus and thus its genetic material has been removed. Now, the reconstructed egg containing the DNA from a donor cell must be treated with chemicals or electric current in order to stimulate cell division. Now, once the cloned embryo reaches a suitable stage, it is transferred to the uterus of a female host where it continues to develop until birth. Now, if we talk about reproductive cloning, dolly or any other animal created using nuclear transfer technology is not truly an identical clone of the donor animal. Only the clone's chromosomal or nuclear DNA is the same as the donor. Some of the clone's genetic materials come from the mitochondria in the cytoplasm of the enucleated egg. Now, mitochondria, which are organelles that serve as power sources to the cell, contain their own short segments of DNA. Acquired mutations in mitochondrial DNA are believed to play an important role in the aging process. And if you talk about therapeutic cloning, it's also called embryo cloning, is the production of human embryos for use in research. Now, the goal of this process is not to create cloned human beings, but rather to harvest stem cells that can be used to study human development and to treat diseases. Now, stem cells are important to biomedical researchers because they can be used to generate virtually any type of specialized cell in the human body. Stem cells are extracted from the egg after it has divided for 5 days, and the egg at this stage of development is called a blastocyst. Now, the extraction process destroys the embryo, which raises a variety of ethical concerns. Now, many researchers hope that one day stem cells can be used to serve as a replacement cell to treat heart disease, Alzheimer's, cancers, and other diseases. Okay, now what are the advantages of cloning? It utilizes good genetics, makes tears into bulls, basically clone for insurance, get another good animal. Now, disadvantages will be obviously ethical concerns. The first thing that will be ethical concerns, obviously expensive. Will the animals after genetical manipulation, will they be normal, natural? Takes many times to clone and can it be dangerous to humans? So if we talk about overview, then we'll start with cells. Cells are the fundamental structure and functional units of living organisms, and cloning describes several different processes of making identical copies of this biological material. Now, why is this important? Because it has the potential to treat a wide range of diseases by generating cell tissues. Now, what is a cell? Cells are the fundamental structure and functional units of living organisms. And eggs and bacteria are made of cells in which the nucleus is surrounded by a membrane. Now, the nucleus contains DNA which provides instructions for the entire organism. We know that. Now, as the cell specializes, only DNA related to the particular functions of the cell remains active. Okay, so obviously the genetic material in all the cells of our body will be same. Somatic cells will be same, but at different locations of the body based on the tissues or the organs, different parts of the DNA might be active. So as a specializes in different parts of the body, only DNA related to that particular function of that cell will remain active. Otherwise, everything else unnecessarily will take a lot of energy that the cell doesn't want. Now, cloning involves making identical copies, right? So cloning can mean several things: to make many identical copies of a DNA molecule or a particular stretch of DNA, to replicate an entire organism, or to produce undifferentiated cells for the purpose of studying and treating diseases, etc. Now, what is recombinant DNA technology? There is a vector, a plasmid backbone, and DNA fragment to be cloned. Then the DNA fragment ligated into vector, creating recombinant DNA molecule. Now, to make many copies of the recombinant DNA molecule, the vector is introduced into bacteria. Now, recombinant DNA technology includes DNA cloning, gene cloning, and molecular cloning. And DNA from one organism is transferred to a bacterial plasmid for replication. So, all the viruses, bacterial artificial chromosomes, and chromosomes may also be used for replicating DNA. Bacterial plasmids are most commonly used in this technology and are called vectors. So this is a brief history of modern cloning. In 1938, Hans proposes animal cloning using somatic cell nuclear transfer. In 52, Robert Briggs and Thomas King transplanted a frog embryo into an unfertilized egg without a nucleus. Then Carl claims to have cloned mice. Uh, in 84, Dever Salter asserts cloning is biologically impossible. Then clones sheep embryos were separating the cells in early embryos, similar to the way twins arise naturally. First verified instance of a cloned mammal. Then at the University of Wisconsin, a cow is cloned from an embryo cow cell using embryo twinning. Then at 1997, a sheep was cloned called Dolly from a mammary breast cell. Now, what is somatic cell nuclear transfer, SCNT? A somatic cell is any cell that is not a germ cell, like it's not an egg or sperm cell, it's a normal cell. Now, somatic cell nuclear transfer involves removing the nucleus from a somatic cell, inserting the nucleus into an egg cell from which the original nucleus has been removed, and uh, shocking the implanted cell, like chemically or electrically, so it will grow and multiply. Now, the new cell is genetically identical to the donor. This technology was first demonstrated in 1983 using amphibians, and this process was attempted with mammals in 1986, but the experiments were limited to developing embryos. Now then later, the first successful mammal clone from an adult somatic cell was a sheep named Dolly. She was produced by implanting an embryo created by SCNT into a mother sheep. Embryonic cells can become any tissue in the body, right? So why do we use cloning technology? Because using the body's own cells to generate tissues not only provides a plentiful supply but also eliminates tissue rejection. Then the technology potentially could be used to treat a wide range of conditions from heart damage to diabetes. Now, what is the difference between therapeutic cloning and reproductive cloning? So, therapeutic cloning yields stem cells that have the potential to repair damaged, disease, or degenerating tissue. It could potentially offer a cure for a wide range of diseases, and there's no tissue rejection problems. Practical clinical applications could take more than 10 years to develop and is aimed at production of stem cells only. Whereas reproductive cloning is not generally condoned by scientists. Its previous cell damage, exposure, UV light transferred to the clone, potential for a wide range of health problems. There is a tech, uh, chance of shortened lifespan, that is, the cells age more quickly. No potential for the treatment of disease, not practical as a solution for fertility problems, and it's aimed at producing a completely new individual. Now, gene cloning, it means amplifying a specific piece of DNA via bacterial cell. Now, cloning vector is a replicating DNA molecule attached to the foreign DNA fragment to be introduced into a cell. It has features that make it easier to insert DNA and select for the presence of vector in cell, the origin of replication, antibody resistance gene, and cloning sites. So if you see, first, a cloning vector must contain an origin of replication recognized in the host cell so that it is replicated along with the DNA that it carries. Second, it should carry selectable markers, traits that enable cells containing the vector to be selected or identified. And third, cloning vector needs a single cleavage site for each of one or more restriction enzymes used. Now, in case of cloning genes, plasmid vectors, linkers, synthetic DNA fragments containing restriction sites, transformation of host cells with plasmids, and selectable markers are used to confirm whether the cells have been transformed or not. So, you see, this is how a plasmid looks. Now, the plasmid and the foreign DNA are cut by the same restriction enzyme, in this case EcoR1, and mix the sticky ends anneal, joining the foreign DNA and plasmid, and then the nicks in the sugar phosphate bonds are sealed by the DNA ligase. This is a conceptual approach, a schematic of how cloning basically works. So basically cloning means you have to insert a foreign piece of DNA of your choice into the plasmid, which will then be inserted into the bacterial cells, and then the bacterial cells will amplify, also amplifying the plasmid in question. So the plasmid and the foreign DNA are cut by the same restriction enzyme, in this case EcoR1, and when mixed, the sticky ends will anneal, joining the foreign DNA and plasmid, and nicks in the sugar phosphate bonds are sealed by DNA ligase. Now, foreign DNA is inserted into the middle of a LAGZ gene. So, we have already discussed blue-white screening in one of our previous classes. So, if the foreign DNA is inserted into the middle of a LAGZ gene, then the bacteria will not be able to produce LAGZ properly, right? So, the LAGZ, it becomes LAGZ negative, okay? And so bacteria will be transformed by the plasmids. Now, bacteria with an intact plasmid produce beta galactosidase, which cleaves X-gal, which is the substrate, and makes the colonies blue. So bacteria with the recombinant plasmid do not synthesize beta galactosidase. The colonies remain white. Because in case of a recombinant plasmid, the LAGZ gene is interrupted by the foreign piece of DNA, right? Otherwise, if it's a normal plasmid, then the LAGZ gene will be intact. In that case, the colonies will be blue. So bacteria without a plasmid will not even grow because there is no antibody resistant gene. So a white colony consists of bacteria carrying a recombinant plasmid, whereas a blue colony means the bacteria carries the normal plasmid where the LAGZ gene is intact and not interrupted by a foreign piece of DNA. This is a bacteria, Platon medium with ampicillin and X-gal. So white colony will consist of bacteria carrying a recombinant plasmid. Now, comparison of plasmids, fast lambda vectors, cosmids, and bacterial adaptation chromosomes. Plasmids are as large as 15 kb, okay, and their method of propagation will be replication, plasmid replication, and their introduction to bacterial transformation. Fast lambda will be as large as 23 kilobase pairs, whereas cosmid as large as 44 kilobase pairs. Both of them, like phage lambda, will go through phage reproduction and phage infection mechanism, whereas cosmid will go through plasmid reproduction and phage infection mechanisms. Whereas bats, which are even huge, like which can carry particles, gene size as large as even 300 kb, they go through plasmid reproduction and can be taken inside cells as electropulsation. Now, for bacterial transformation, we can use modified strain of E. coli, because in this case then the restriction system will be knocked out and the recombination system will be limited and LPS will be truncated. So pores will be made in the cell membrane by treatment with calcium chloride. Then cells will be taken in, must be kept cold to prevent pores from closing. Then there will be a gentle heat shock that will push the plasmid inside. Now DNA added to cells forms intimate interaction with membrane. So briefly it is moved to 42 degrees Celsius heat shock. Then it will give them stress and cause them to take up DNA. Now cloning is actually a loaded term that can be used to mean very different things. So cutting a piece of DNA from one organism and inserting it into a vector where it can be replicated by a host organism. Sometimes it is called subcloning because only part of the organism's DNA is being cloned. Now using nuclear DNA from one organism to create a second organism with the same nuclear DNA. Now, restriction enzymes are proteins that cleaves DNA molecules at specific sites, producing discrete fragments of DNA. Now, why would bacterial cells contain proteins that cleave DNA at specific sequences? So, generally, restriction enzymes are thought to protect bacterial cells from phage or bacterial virus infection. So, bacterial cells that contain restriction enzymes can cut up invasive viral DNA without damaging their host DNA. So this is again a schematic in 1972, schematic in the sense timeline. Paul Berg and colleagues made the first artificial recombinant DNA molecule, and they demonstrated that DNA of simian virus 40 could be linearized by EcoAD1. So they created a circular dimer of simian virus DNA by joining two linearized fragments. Also inserted pieces of lambda phage DNA into linearized simian 40 virus molecule. Now, Herbert Boyd and Stanley Cohen built on the work of Berg, Nathan, and Smith to use a restriction enzyme to isolate a single gene, place it into a plasmid vector. Now, bacterial cells were then transformed into with the recombinant plasmid. So, the bacteria host cells replicating the plasmid, producing any copies of the gene, thus amplifying it. So, the practical application was that expensive human protein products like insulin, which we use to treat disease, could eventually be produced from recombinant molecules in the laboratory using bacteria or another host, right? So there will be no scarcity of insulin if needed be. So human protein products like insulin could be used in very large quantities from the recombinant molecule. Patients no longer had to use insulin isolated from pigs or cows. Now, plasmids are circular pieces of DNA found naturally in bacteria, so they can carry antibiotic resistance genes, genes for receptors, toxins, or other proteins, and they replicate separately from the genome of the organism. So, they can be engineered to be useful cloning vectors. Now, plasmid vectors can be designed with a variety of features: antibiotic resistance, colorimetric markers, strong or weak promoters for driving expression of a protein. So, this is an antibiotic resistant markers. This is a multiple cloning site. The cloning of a piece of DNA. Now, if you have to cut the plasmid vector with one endonuclease, and then you have to cut the DNA of insert from the source using a same endonuclease, and then you can ligate the insert of interest into the cut plasmid. That's how it works. So, for restriction digestion, They are all specific conditions under which they work best. Some of the conditions that must be considered when performing the restriction digest are temperature, salt concentration, and the purity of the DNA. Very important. So the insert of interest that you want to clone into your plasmid needs to be separated from the other DNA, right? So you can separate your fragment using gel electrophoresis. You can then purify the DNA from the gel by cutting the band out of the gel and then using a variety of techniques to separate DNA from the gel matrix. Now ligation is the basically gluing process. It's called molecular glue. Ligases are called molecular glue. They are help in joining two pieces of DNA from different zones together through the formation of a covalent bond. Now DNA ligase is the enzyme used to catalyze this reaction, and DNA ligation requires ATP. Now once you have the plasmid ready, insert recombinant plasmid ready, you have to insert into the bacteria. So you will need to insert it into a bacterial host cell so that it can be replicated. So the process of introducing the foreign DNA into the bacterial cell is called transformation. Now not every bacterial cell is able to take up plasmid DNA. Bacterial cells that can take up DNA from the environment are said to be competent. Now they can treat cells with electric current or divalent cations to increase the likelihood that DNA will be taken up. Then two methods of transforming: heat shock and electroporation. Now the transformed bacterial cells are grown on selective media containing antibiotic to select for cells that took up the plasmid. Now for blue white screening to determine if the plasmid contains an insert, the transformants are grown on plates containing XGAL and IPTG. IPTG basically induces the production of the particular protein. XGAL is the substrate. So if XGAL is broken into something blue, that means the recombinant plasmid is not there. Whereas if the XGAL, there is no effect on the XGAL and the you can see white colonies, that means there is a recombinant plasmid vector. Okay. So cells can take, if you are trying to transform, cells can take either plasmid only, which plasmids do not have any insert, plasmid with insert, clone, foreign data from the environment, or nothing at all. So even if your plasmid contains your insert, it may not be able to generate functional proteins from your clone DNA. So this gene may not be intact, or mutations could have been introduced that disrupt it, and the protein encoded by the gene may require post-translational modifications to function. Also, some enzymes are complex of peptides expressed from separate genes. Now, the expression of a clone gene can be accomplished by the E. coli host mammalian cells. If the plasmid is designed for expression in mammalian cells using an in vitro using cell-free system, relationship between genes and the proteins. Now, molecular cloning, as we have already discussed, clone means to have identical copies. DNA cloning involves separating a specific gene or segment from large chromosome, attaching to a small carrier DNA. The resultant hybrid DNA is called recombinant DNA, which is transferred to a proper host and replicated to make multiple copies of the selected gene. Now, when cloned under an appropriate expression vector, a gene can be expressed, that is, can be transcribed and translated, and whatever it was coding will come out as a protein at desired level to produce recombinant proteins. Now, this technology has made it possible to isolate, clone, and produce DNA for all the genes in appropriate quantity so that they can be sequenced and characterized. Similarly, some of the genes which are expressed at very low level can be cloned and desired amounts of recombinant proteins can be produced. So this is the keratic chromosome, cloning vector, plasmid cleavage with restriction endonucleases, separation of fragments, all of these ultimately DNA ligase and cell propagation. These are some of the enzymes used in recombinant DNA technology. I think we have already discussed in one of the previous classes. There are restriction endonucleases, there are polymerases, ligases, Reverse transcription polynucleotide kinase is terminal transferase, exonuclease 3, bacteriophage, lambda, exonuclease, alkaline phosphate, etc. Now, circular plasmid DNAs are the most common cloning vectors. They are around 1 to 200 kb along a long DNA duplexes of containing required genetic material, and this replication origin to permit their astronomers production in the host cell. Now, the plasmid vectors contain specific genes responsible for antibody resistance as well, which can be used to select the bacterial contains containing recombinant plasmids. So these plasmids have antibody resistance, probably for example ampicillin resistance. So even in a media with ampicillin, these will not die. In order to clone the foreign DNA at a specific site, a synthetic oligonucleotide containing restriction sites for several is ligated in the plasmid. This region is referred as polylinker region. For joining of the two fragments, TTT is a mammalian enzyme which adds a nucleotide to the 3' OH group of DNA without any requirement of primer, and the two DNA fragments to be joined and subjected to TTT reaction. The balance of DTDP and DATP separately to add polylysine to one DNA. The two DNA fragments with cohesive ends are annealed. The gaps are filled with DNA polymerases, and then they're just joining covalently by ligase. So TTT requires at least the 3 nucleotides free at 3' end and can be created by bacteriophage lambda exonuclease. I've talked about most of these in one of the previous classes. Will not go into very much detail in the interest of time. I've talked about all these enzymes and all of these. Go into details. So coming back to the question that we started with, the molar ratio of vector to insert for ligation reaction can be many. It can be 1:10 as well, depending on the number of recombinant vectors we are actually getting, but the most probable number is 1:3. Okay. If you have any doubts in this question, we can discuss. I'll wait for 5 minutes and we'll move on to the next question. Okay. So, moving on to the next question for today. So, the next question is, which of the following proteins helps the unwinding of DNA helix during DNA replication? Okay. Okay, one second. One second. Okay, so now you can see the answer over here. Okay, one second. Let me. Already seen the answer, but anyway. Which of the following protein helps the unwinding of DNA helix during DNA replication? Helicase, DNA ligase, dithering proteins, or SSB? SSB are basically single strand binding proteins, basically. So, which one do you think will be right?
Option helicase.
Option?
A
Right,
helicase.
right, it's helicase. So if you see it's the structure of the DNA we know it's a double helix, basically with hydrogen bonding. G and C, or guanine and cytosine will have um, guanine and cytosine will have three hydrogen bonds, and AT will have two. You know it's a double helix that are held together by weak hydrogen bonds. Okay, and they are basically nucleotides, that are subunits that make up the DNA. Okay, they are made up of three components: a phosphate group, a deoxyribose, the 5-carbon sugar, and a nitrogen containing base, right? So in case of DNA structure, it has 4 nitrogen bases: purines and pyrimidine. Among purines we have the adenine and guanine. In the pyrimidines we have the thymine and the cytosine, right? So this is the DNA structure. These are helicases. Basically, helicases unwind the DNA structure. Otherwise, the DNA structure is a compact double helix, but the helicases unwind the DNA structure, and this position is known as replication fork. Now, purines on one strand always pair with pyrimidines on the other strand, like adenine pairs with thymine always, guanine pairs with cytosine always, and in case of complementary strands, like sequence of bases on one strand determines sequence on the other strand. Because we know if there is A on one strand, at that position it will be T on the other strand, right? So basically that is what we mean by complementary strands. So make up the double helix and serve as a template to build DNA. This is a pairing between the bases. Now for DNA replication, step 1 means double helix unwinds DNA helicase, sorry. Then, and it's basically an enzyme that breaks apart the hydrogen bonds, right? Then we have the hydrogen bond. So these bonds hold the nucleotides together, and proteins, which is obviously attached to the strands to keep the bonds from reforming. Okay. So SSP, or single strand binding proteins, can be a type of proteins that are attached to the strands to keep the bonds from reforming. Then we have the replication forks, two areas where the DNA opens up to allow replication to occur. Now, DNA replication, in which nucleotides are added. They have DNA polymerase enzyme that moves along each strand adding nucleotides. And the step 3 will be 2 strands of DNA are formed. So DNA polymerase will remain attached until all the DNA is replicated, and the new DNA, once process is completed, 2 new strands are completed. A nucleotide sequence, identical sequences will be present on both the strands. Now what will you do for checking the errors? Proofreading. First of all, you have one strand. The complementary strand will be formed with the help of polymerase, right? But there might be some errors. Not everything can be completely perfect, right? So there is a proofreading facility as well. So polymerase can add only a nucleotide if the previous one was correctly placed. So most polymerases have a proofreading exonuclease activity as well. That is backtracking. DNA can go back and remove, the enzyme can go back and remove the incorrect nucleotide, and replace it with the correct one. Okay, so some information about DNA replication. A double helix separate into two single strands, and each strand serves as a template, right? Template on which complementary strand is synthesized. Its a mechanism is required to separate the strands locally for replication. A mechanism is required to release the strain created by local unwinding, and the mechanism is required to account for the high fidelity of duplication. So the mechanism is required to account for the speed of replication as well. 2000 nucleotide per second in E. coli. So these are different structures of DNA. It can be A, B, or Z. One second. Okay, so these are different types of DNA double helix structures. Uh, okay, so if you can see in different cases, the major and the minor groups vary, right? So we know about semi-conservative replication. Okay, so this is the original parent molecule, and then the strands separate, and each has a new strand being formed. So these are the first generation daughter molecules. Not going into too much detail. So if you see, this is E. coli DNA B helicase. So the helicase will translocate along double-stranded DNA from 3' to 5' direction. So we can do helicase assays as well to showcase that. This is structure of bacterial helicase. So for DNA duplex to replicate, the two strands must be separated from each other, at least locally. Helicase will use ATP energy to perform this strand separation job. Both A1 and B1 will bind to single-stranded DNA. ATP hydrolysis leads to cleft closure and sliding of single-stranded DNA. Now, since A1 has a tighter grip of ssDNA, this causes a net translocation of the enzyme towards the double-stranded DNA. Okay. So we'll not go into too much detail. These are the overview of replication, detailed view of the replication fork. So these people are known for their discovery of mechanisms in the biological synthesis of ribonucleic acid and deoxyribonucleic acid. So we know that DNA replication is the process of making an identical copy of section of duplex DNA using existing DNA as a template for the synthesis of new DNA strands. And in humans and other eukaryotes, replication occurs in the cell nucleus, right? So what are the possible models of DNA replication? Delbruck suggested that Watson and Crick model of DNA could theoretically replicate by 3 modes. Okay. So what are the major enzymes and proteins involved in DNA replication? That is polymerase 1. Okay. Discovered by Arthur Kornberg. There is a 5' to 3' polymerization. That is synthesis of the polymers from monomers in one direction, 5' to 3'. 3' to 5', the exonuclease activity or the proofreading activity comes into play. So hydrolysis of single nucleotides from the end of a DNA or RNA chain. It functions as a proofreader, thus acts in repair synthesis. 5' to 3' exonuclease activity is there. It removes thymine dimers. As it moves ahead, it cuts off ribonucleotides in front and adds deoxyribonucleotides behind. Excuse me. For polymerase 2, we have the 5' to 3' polymerization activity, And 3 to 5 exonuclease activity involves in editing, in repair, application of UV damage. Also, it can elongate Okazaki fragments in absence of polymerase 1. Okay, polymerase 2 is a very complex enzyme. It is associated with 9 other proteins in its active form. The core enzyme, the smallest aggregate having function of polymerase, 5' to 3' polymerization and 3' to 5' exonuclease. Now, DNA origin binding protein binds to origin of replication just before initiation. DNA B or helicase unwinds DNA strands using ATP energy, and single strand binding protein will be binds to and stabilizes unwound single stranded DNA. And DNA serially forms a complex with helicase to load and function on DNA template. So, this entire unit consisting of enzymes, template strand, and newly synthesized strand at the replication fork is called replicosome. Okay, so coming back to the question that we started with, the answer will be helicase. Which of the following protein helps in the unwinding of DNA helix during DNA replication? That's helicase. Okay, we'll wait here for 2-3 minutes and then we can move on to the next question. So moving on to the next question for today. The removal of the terminal phosphate groups from DNA is done by A. Exonuclease, B. Restriction endonuclease, C is alkaline phosphatase, and D is DNA ligase. Which one do you think is the correct answer?
Ma'am, answer is already given.
Sorry, I actually forgot to put the animation. But do you have any other opinions? Can it be anything other than alkaline phosphatase? If yes, I wanted to know.
No, ma'am, because DNA ligase is somehow joining,
Correct.
so
It's a molecular
it
glue.
will not remove. And other option like exonuclease is also cutting, but it doesn't remove any
Correct.
phosphate group
Correct.
from the
Correct.
DNA.
So both
And restriction endonuclease also like
cutting
they are
only, but yeah,
they are from the nucleus class,
correct.
so they don't remove any phosphate group from the
Right.
DNA.
So both exonucleases and Russian endonucleases, both of these are nucleus category. One of exonucleus can cut from any ends, whereas endonuclease means it can cut in the middle as well, and at very specific sites. DNA ligase is called a molecular glue, so definitely not something that will cut anything out. Alkaline phosphatase. So phosphatase, whenever you will see kinase and phosphatase, these two enzymes have opposite activities. When we talk about kinase, we should have in mind that there is something that is adding phosphate group to something. And when we say phosphatase, that is, it is removing the phosphate group from something. Okay. So if we talk about the principles of cloning, vector and cloning strategies, we have talked about all of these. So DNA cloning is a technique for reproducing DNA fragments. It can be achieved by two different processes, cell-based or using PCR. Now a vector is required to carry the DNA fragment of interest into the host cell that we know. And DNA cloning allows a copy of any specific part of a DNA to be selected among many others and reproduced in an unlimited amount, right? So this technique is the first stage of most of the genetic engineering experiments, production of DNA libraries, PCR, DNA sequencing, etc. Now, DNA cloning means massive amplification of DNA sequences, stable propagation of DNA sequences. A single DNA molecule can be amplified, allowing it to be studied or sequenced, manipulated, which is mutagenized or engineered, expressed generation of, that is means generation of protein, right? When we say studied, basically we do the sequencing of the segment. Manipulated means basically engineered, like we manipulate it according to our own needs. We can add or delete anything. And express basically means the generation of protein, right? Give me a second. Okay, so cloning process means gene of interest is cut out with Russian endonuclease. The host plasmid is cut with same RE, and gene is inserted into plasmid and ligated with the ligase. Now new plasmid is inserted into the bacterium. That is, transformation is carried out. Now plasmid cloning strategy, it involves 5 steps. Enzyme restriction, digest of DNA sample, Inhibition digest of DNA plasmid vector, ligation of DNA sample products and plasmid vector, and transformation with the ligation products. Now, and growth on agar plates with selection for antibiotic resistance. Now, for example, the first step will be redirection of DNA sample, right? So, whatever is the gene of interest, we'll digest the DNA sample with a known restriction endonuclease. For example, EcoR1. Known by the term known, I means endonuclease whose sites are known to us, whose restriction sites are known to us. Right? Then you digest the plasmid DNA also using the same enzyme at the MCS region. Then you ligate the DNA sample and the plasmid DNA, right? You basically combine all of these at DNA ligase and ATP. Okay. Now then we'll transform the ligated products. Now that the ligation has happened, so plasmid and DNA segment will be together. So that will be transformed together. So the process of transferring exogenous DNA into cells is called transformation. So there are basically two general methods for transforming bacteria. The first is the chemical method utilizing calcium chloride and heat shock to promote DNA entry into cells. And the second method is called electroporation based on a short pulse of electric charge to facilitate DNA uptake. Now for chemical transformation with calcium chloride, we should have a culture in the log phase that you will centrifuge. You suspend bacterial pellet in calcium chloride solution. You will then chill on ice, and first of all you will make competent cells. Competent cells will be more prone and more susceptible to the plasmid DNA that will be taken inside. Normal cells might not take it, and that is how we make the competent cells using calcium chloride only. We basically make pores on the cell surface, cell membrane, and then we chill it on ice with the plasmids, and then we do heat shock for 1.5 minutes with the plasmid of interest, and that's how we do the transformation. And then we plate on LB and ampicillin. Ampicillin because the plasmid of interest will have the ampicillin resistance gene. Okay, we can also do transformation by electroporation. That is comparatively, I think the calcium chloride and heat shock method is more popular in labs. Okay, so electroporation technique can also be used. Basically, you give an electroshock in saline buffer and the plasmid is taken inside. Then you grow on agar plates. Now, how will you separate? How will you differentiate if the bacteria has taken in a plasmid or not? So your plasmid will have an antibiotic resistance gene. So your plasmids, if taken inside by the cell, only then the cells will survive on the antibiotic plate. Otherwise, the cells will survive. So that's one technique to ensure that your plasmid has gone inside. But now, what if only the plasmid has gone inside? Your recombinant plasmid has not gone inside. Only the empty plasmid has gone inside. Your insert containing plasmid has not gone inside. For that, we do blue white screening basically. So that exgal is a type of substrate that we, and ampicillin, we add it into the agar only. And this is our vector. Okay, this is our multiple cloning site and our LAGZ gene. So if you see, the insert is not here, so everything is fine. So the LAGZ gene is also functional. So it gives rise to a beta galactosidase which converts exgal to blue colonies. So you see, it's all blue. But here if you see, the insert is over here, right? So the insert has disrupted the LAGZ segment. So your LAGZ gene is not capable of producing a functional beta galactosidase. So there is no um, changing of the X gal to a blue colony. Okay, so that will give to rise to white colonies. Yeah, so there will be some plasmids in the on the plate which will have the insert, some will not have the insert. So only the plasmids containing the insert will give rise to white colonies, and the plasmids not containing the insert will give rise to blue colonies, right? And if there is self-ligation, that is the insert has self-ligated and there is no plasmid, then those type of bacteria will not grow on these plates only, because if there is no plasmid, there is no antibiotic resistance gene, this is not there. So the bacteria will die on exposure to ampicillin. That is how the screening process are carried out. As I was saying, blue colonies will represent ampicillin resistant bacteria that contain P vector and express a functional alpha fragment from an intact LAGZ alpha coding sequence, whereas white colonies will represent ampicillin resistant bacteria that contain the insert and do not produce a LAGZ alpha fragment. So these are some of the terms used in cloning. DNA recombination we have talked about. DNA fragment to be cloned is inserted into the vector. Transformation, obviously the recombinant DNA will enter into the host cell and proliferate. Sorry. Selective amplification is a specific antibiotic is added to kill E. coli without any protection. The transformed E. coli is protected by the antibiotic resistance gene. AMPR that we saw in the last case. An isolation of desired DNA clones. Now, cloning vectors are DNA molecules that are used to transport clone sequences between biological hosts and the test tube. Okay, so cloning vectors share four common properties: ability to promote autonomous replication, it can contain a genetic marker as well for selection, unique restriction size to facilitate cloning of insert DNA, and a minimum amount of non-essential DNA to optimize cloning. Okay, 1 second. So bacterial cells may contain extra chromosomal DNA called plasmids, and plasmids are usually represented by small circular DNA. And some plasmids are present in multiple copies in the cell. We have talked about plasmid vectors as well. They have replication origin. They have a gene that permits selection, and the selective gene is AMPR. It encodes the enzyme beta lactamase, which inactivates ampicillin around. And exogenous DNA can be inserted into the bracketed region as well. Selectable marker, we have talked about this also, is required for the maintenance of plasmid inside the cell. Because of the presence of selectable marker, the plasmid becomes useful for the cell. So under the selective conditions, only cells that contain plasmids with selectable marker can survive, and genes that confer resistance to various antibiotics are used. So genes that make cells resistant to ampicillin, neomycin or chlorphenicol are used. We know about origin of replication as well. Basically, it's a DNA segment replaced by the cellular DNA replication enzymes, and without the replication origin, DNA cannot be replicated within the cell. Then we have the MCS on the multiple cloning site. Basically, it contains MCS or polylinker, a DNA segment with several unique sites for restriction endonucleases located next to each other. The external sites of the polylinker are not present anywhere else in the plasmid, and cutting plasmids with one of the restriction sites that recognizes site in the polylinker does not disrupt any of the essential features of the vector. Now, gene to be cloned can be introduced into the cloning vector at one of the restriction sites present in the polylinker. Okay. We have talked about the type of cloning vectors. It can be plasmid vectors. It can be, what are the disadvantages? It cannot accept large fragments, and standard methods of transmission might be inefficient. We have the bacteriophage lambda vector, which is a bacterial virus basically that uses E. coli as host. Its structure is that of a typical Faj. The lambda viral genome is huge, 48.5 kb linear DNA with 12 base single-stranded DNA sticky ends at both ends. These ends are complementary in sequence and can hybridize to each other. Infection, lambda tail fibers absorb to a cell surface receptor. Tail contracts and the DNA is injected, and the DNA circularizes at the cause site, and lambda begins its life cycle in the E. coli host. We have the cosmid vector. It can clone large inserts of DNA, size 45 kb. These are plasmids with one or two lambda cos sites, and presence of the cos site permits in vitro packaging of cosmid DNA into lambda particles. So they have some advantages of lambda as cloning vehicle. The strong selection for cloning of large inserts and infection process rather than transformation for entry of chimeric DNA into the E. coli host. So they can maintain cosmids as sparse particles in solution. But cosmids are plasmids, thus do not form plaques, but rather cloning proceeds via E. coli colony formation. Then we have the BAC and the YAC. As I said, BAC will be bacterial artificial chromosome and YAC will be yeast artificial chromosome. So they are specialized linear DNA vectors that resemble normal yeast chromosomes, right? And they contain telomeres that stabilize chromosome ends, centromere that ensures chromosome partitioning between two daughter cells, and a selective marker gene. So they can carry hundreds or thousands of base pairs of foreign DNA. So their purpose is cloning vehicles that propagate in eukaryotic cell hosts as eukaryotic chromosomes. They can carry very large inserts, and the YAC cloning vehicles are plasmids, and final chimeric DNA is a linear DNA molecule with telomeric ends. Okay, so they often have a selection for an insert, and the YAC cloning vehicles often have a bacterial origin of DNA replication and a selectable marker for propagation of the YAC through bacteria. And the YAC can use both yeast and bacteria as a host. Now, if we talk about PACs and BACs, PAC is P1 derived artificial chromosomes and BAC is bacterial artificial chromosomes. So in PAC, we have the E. coli bacteriophage is similar to fast lambda in that it can exist in E. coli in a prophage state. It exists in E. coli cell as a plasmid, not integrated into the E. coli chromosome. Okay, so it's like an episome, it's not integrated into the genome. Then P1 cloning vehicles have been constructed that permit cloning of large DNA fragments, few hundred kilobase pairs of DNA, and cloning and propagation of the chimeric DNA as a P1 plasmid inside the E. coli cells. For BAC, these chimeric DNA molecules use a naturally occurring low copy numbered bacterial plasmid origin of replication, such as that of F plasmid in E. coli, and they can be cloned as plasmid into bacterial host, and its natural stability generally permits cloning of large pieces of insert DNA up to few hundred kilobase pairs of DNA. Then we have the retroviral vectors. They're used to introduce new or altered genes into the genomes of human and animal cells. They're RNA viruses. The viral RNA is converted into DNA by the viral reverse transcriptase and then is efficiently integrated into the host genome. Any foreign or mutated host gene introduced into the retroviral genome will be integrated into the host chromosome and can reside there practically indefinitely. So retroviral vectors are widely used to study oncogenes and other human genes. So in case of expression vectors, they will allow a clone segment of DNA to be translated into protein inside a bacterial eukaryotic cell. So vectors will contain the in vivo promoter, ampicillin selection, and sequencing primers. They can produce large amounts of a specific protein, permit studies of the structure and function of proteins, and can be useful when proteins are rare cellular components or difficult to isolate. Okay, but what are the common problems with bacterial expression systems? There's low expression levels, promoter chain, plasmid change can be done, cell type also can be changed, and rare tRNAs can also be done for rare codons on second plasmid because of the fact of codon optimization. Or there can be severe protein degradation, like we can use proteasome inhibitors and other protease inhibitors to counter that, or try induction at lower temperature. There can be missing translational modification as well if we have to co-express with kinases. Glycolization will not be carried out also because yeast or mammalian expression system are only compatible with that. There might be misfolded protein, like the protein is produced but it's misfolded, so it's functionally not relevant as well. So you can express with a co-express with the GROW-L, which is chaperone, which helps in correct folding of the protein. Now, what are reporter gene vectors? A gene that encodes a protein whose activity can be easily assayed in a cell in a visual readout. in which it is not normally expressed. So these genes are linked to regulatory sequences whose function is being tested, and changes in transcriptional activity from the regulatory sequences are detected by changes in the level of reporter gene expression. Then we can talk about shuttle vectors can replicate in two different organisms, bacteria and yeast, or mammalian cells and bacteria. They are the appropriate origins of replication, hence one can clone a gene in bacteria, maybe modify it or mutate it in bacteria and test its function by introducing it into yeast or animal cells. What is the cloning strategy? Strategy depends on the starting information and the desired endpoint. Now, how are genes cloned using plasmids? To understand how genes are cloned, we introduce three terms: recombinant DNA, vector, and plasmids. Okay, we've talked about all of these. So, why plasmids are good cloning vectors? Because of small size, circularity, replication independent of host cell, several copies may present, and frequently can have antibiotic resistance. We have talked about this, and as we know, cloning method also we have talked about. Blue wire reading we have talked about. So you see laxine is not, if laxine is not expressed considerably, XGAL does not activate gene expression. So we have to use IPDG also as an inducer. Okay. So if you talk about alkaline phosphatase, which is what the question was actually about. See, these are the basic functions of these different enzymes that we use in molecular biology. Alkaline phosphatase removes phosphate groups from 5' ends of DNA. It prevents the unwanted religation of cut DNA. Then DNA ligase joins compatible ends of DNA fragments. Blunt, or complementary cohesive ends. It uses ADP. The enzymes most used in molecular biology labs cut within the recognition sites and generate one of three different types of ends. So they can have 3' overhangs. We see asymmetrical cutting within the recognition site where the result is a single stranded overhang from the two 3' ends. For example, KPN bond cuts in this manner. Then we have blunt ends, and then they cut at precisely opposite sides in the two strands of DNA generate blunt ends without overhangs. SMA1 is an example of an enzyme that generates blunt ends. And we have can actually convert a 5' overhang to a blunt end. So both Tleno and T4 DNA polymerase can be used to fill in 5' protruding ends with DNTPs. It is used in joining DNA fragments with incompatible ends. So once the ends have been blunted, ligation can proceed. You can convert a 3' overhang as well to a blunt end. T4 DNA polymerase has a 3' to 5' exonuclease activity. In the presence of excess DNTPs, it will convert a 3' protruding end to a blunt end, and ligation can then proceed. Now, if you talk about directional coding, often one desires to insert foreign DNA in a particular orientation. So this can be done by making two cleavages with two different restriction enzymes. So you can construct foreign DNA with same restriction enzymes, and foreign DNA can only be inserted in one direction. So good efficiency of ligation of foreign DNA into a vector can be achieved if both the vector and the insert DNA are cut with two different restriction enzymes. Then it will be forming a direction. It's called directional cloning, which will leave single stranded ends, right, cohesive ends, and the DNA is ligated in only one direction, and there is only a low background of non-recombinant plasmids. So if only one restriction enzyme is used to cut the vector and the insert, then efficiency of ligation is lower. DNA can be inserted in two directions and tandem copies of inserts may be found. So to avoid high background of non-recombinants, alkaline phosphatase is used to remove 5' phosphate groups from the cut vector to prevent self-ligation. So alkaline phosphatase removes 5' phosphate groups from DNA and RNA. It will also remove phosphates from nucleotides and proteins, and these enzymes are most active at alkaline pH. So there are two primary uses for alkaline phosphatase in DNA manipulations. You can remove 5' phosphates from plasmid and bacteriophage vectors that have been cut with the restriction enzyme. In subsequent ligation reactions, this treatment prevents self-ligation of the vector and thereby greatly facilitates ligation of other DNA fragments into the vector. Examples of cloning. Then if we talk about removing the 5' phosphates from the fragments of DNA prior to labeling with radioactive phosphate, polynucleotide kinase is much more effective in phosphorylating DNA if the 5' phosphate has previously been removed. So if you have blunt ends, it's actually difficult for cloning. So you need high concentration of DNA and ligases. Okay. If you have different protruding termini, that requires purification of plasmid after digestion. And if you have identical protruding terminal, then phosphatase treatment of the linearized plasmid vector works. Talked about all of these. If we talk about restriction mapping, basically it is determining the order of restriction sites in a clone fragment. Basically cut the fragment using the restriction enzyme and then run it on a gel. And basically which gel it refers, it means separating DNA fragments by molecular weight. Southern blot analysis is also quite famous. It is DNA is transferred or blotted to filter paper, and that is exposed to a DNA probe. It binds specifically to target DNA immobilized on filter. Then we have DNA sequencing. It provides complete order of bases in a DNA fragment. So you can see if it is in the correct orientation based on the start and the stop codon, it will give a 200 base pairs. But if it is the wrong orientation, it will actually give a 600 base pairs. That is how you can check the orientation as well. So what are the different forms in of DNA in a plasmid? Uncut plasmid can be in any of the 5 forms, mostly 3, but you can even segregate it further. Nicked circular, if you see this is a nicked form, then linear, then covalently bonded, supercoil, and circular single stranded. Okay, the one which is the most compact will run the fastest, right? So the exact distances between the bands of these different forms is influenced by the percentage of agarose. Obviously, if the percentage of agarose is more, then the pore size will be very small, so it will take more time for these segments to pass through. Obviously, time of electrophoresis, if you allow them more time, there will be better resolution. Degree of supercoiling, more the supercoiling, more compact the structure, more easily it will pass through. Size of the DNA, obviously, size, if size is small, it will move, it will go to the towards the positive terminal very fast. Okay, but all of these. For all of these. So phosphatase catalyzes the cleavage of a phosphate group from substrate by using water molecule. It undergoes hydrolytic cleavage, and this reaction is not reversible. So on the basis of their activity, there are two types of phosphatases: acid phosphatase and alkaline phosphatase. In both forms, the alkaline phosphatase are most common. Okay, especially there's a phosphatase that remove a phosphate group from protein are called phosphoprotein phosphatase. This is schematic representation of a hydrolytic cleavage of a phosphate group. What is a deoxyribonuclease or DNase? It's a nuclease enzyme that can catalyze the hydrolytic cleavage of phosphodiester bonds in the DNA backbone. Okay, it's based on the position of action. These enzymes are broadly classified as endodeoxyribonuclease and exodeoxyribonuclease. Okay, endo will cleave DNA sequence internally and exo will cleave the terminal nucleotides. Now, unlike restriction enzymes, DNA does not have any specific restriction or restriction site and cleave DNA sequence at random locations. Methylase or methyltransferase catalyzes the transfer of methyl group to the substrate, and the process of transfer of methyl group to the substrate is called methylation. So it's a common phenomenon in DNA and protein structure, and these use a reactive methyl group that is bound to sulfurin as adenosyl methionine, which acts as the methyl donor. So methylation normally occurs on cytosine residue in DNA sequence, and in protein methylation occurs on the nitrogen atom either on the N terminus or on the side chain of protein. So DNA methylation regulates gene or silences gene without changing DNA sequence is the part of epigenetic regulation. So in bacterial system, methylation plays a major role in preventing their genome from degradation by restriction enzymes. It is a part of restriction modification system in bacteria. If we talk about ligases, they catalyze the formation of phosphodiester bond between two deoxynucleotide residues of two DNA strands. So DNA ligase enzyme requires a free hydroxyl group at the 3' end of the DNA and a phosphate group at the 5' end of the other end. Requires energy in the process. The role of DNA ligase is to seal nicks in the backbone of double-stranded DNA after lacking strand formation to join the Okazaki fragments. So this joining process is essential for the normal synthesis of DNA and for repairing damaged DNA. It has been exploited by genetic engineers to join DNA chains to form recombinant DNA molecules. Usually single-stranded DNA breaks are repaired using the complementary strand as a template, but sometimes double-stranded DNA breaks can also be repaired with the help of DNA ligase. So coming back to the question that we started with, the removal of the terminal phosphate groups from DNA is done by alkaline phosphatase.
Ma'am,
Ma'am,
Yes.
suppose we are having a plasmid with two antibiotic resistance genes, tetracycline and ampicillin. If we cut at the ampicillin resistant gene, so that ampicillin, the effect of ampicillin will be not present.
Okay.
So when we are growing in a medium of ampicillin, that bacteria containing, so the norm which do not have that resistant gene, they will die. So how we can differentiate at that time?
So if you have another, so you need to keep your antibiotic resistance gene intact for that case. So if you have both ampicillin and tetracycline, then probably you can use media. I have not. There should be people using, but ampicillin, kanamycin, hygromycin, these are the most prominent, right? Popular, like these people have done in, I've heard people doing this in labs, but theoretically tetracycline can also be mixed with agar and the bacteria can be plated. If your ampicillin is disrupted in the middle, it will not show any ampicillin resistance. Okay, so that bacteria, even though it contains the ampicillin resistance gene, just because it has been cut by something and an extra something has been added to it, it will not show ampicillin resistance. So yes, it will die in ampicillin media, right? But if you have an agar plate with tetracycline, the tetracycline resistance gene, if that is kept proper and that is not disrupted, then that should work. So whatever bacteria have the tetracycline resistance gene site intact will produce that particular protein that will help in disrupting tetracycline in the media, and thus they will survive. So if you have another antibiotic resistance gene, you don't have to worry. You can use that.
Ma'am, but if there is one resistance in,
And then you cannot do this. That's what I say for blue white staining. The MCS is generally not MCS. You have to cut in the MCS region. The antibiotic resistance gene is generally not coinciding with the MCS region. The MCS is a multiple cloning site, right? So the multiple cloning site will have restriction sites of various enzymes. So plasmids are made like that so that you can use a variety of enzymes depending on your purpose and the gene of interest. So if you cut the antibiotic resistance gene, then you will not be able to uh, differentiate it on agar plates, right? So that cannot be done. So if you disrupt the antibiotic resistance gene, then none of the bacteria will grow. It will not grow. So you can grow it in an agar plate without any antibiotic. Then it will grow. But then you will not be able to figure out whether your for example, if you're putting your gene of insert in the antibiotic resistance gene region, right? So, and then you're growing it in an agar plate without any antibiotic, then the plasmid without your gene of interest will also grow because there's no antibiotic, so it doesn't have to grow, and the bacteria in which your antibiotic resistance gene has been disrupted by a target gene, that will also grow, right, in a normal agar plate. So you will not be able to differentiate at this with this experiment how which one has your gene of interest. Now if you take a plate with agar with ampicillin, then your bacteria that will not have the gene will grow, because in that bacteria the ampicillin is not disrupted. But the other bacteria in which you have disrupted the ampicillin with the gene of interest, that will not grow only. So if that is not growing, then how will you move ahead with the further experiments? You will not be able to, right? So your antibiotic resistance gene, if there is only one in the plasmid, it is generally kept away from the MCS. You do not generally uh, do anything at the ampicillin at the antibiotic resistance gene, because that is a selectable marker that helps you figure out if your bacteria has the plasmid with the gene of bacteria has a plasmid or not, right? So that is how it works.
But ma'am, when it is cutting in an MCS region, it is beyond. Suppose that plasmid is that DNA is not entering. How could we know that?
DNA is not entering, matlab you, so you, in the MCS region you will cut with two different enzymes. For example, the way I talked for directional cloning, and for insert gene also you will cut at the two ends with two different enzymes. So you will know if it is ligating properly, it will ligate in a directional manner. Okay, and this MCS region will get your gene of interest inserted, but the antibiotic resistance part will not be hampered. That is the only way you can segregate, differentiate on agar plates. Otherwise, you have to take the bacterial clones and go for sequencing. Sequencing is the sure shot method that your gene of. So what whenever we do in labs also we do cloning and everything, right? We we sequence it even after that. Okay, so cloning is, by cloning we can figure out by tomorrow or the day after. Sequencing we do like whenever we're handling or anything we are doing, we have to ensure that there are no mutations taking place, random rampant mutations happening at that point. So we anyway sequence our clones at the end. But the first step is by agar ampicillin screening. So by that we know that the bacteria which will not have the plasmid, will not have the ampicillin resistance, will die. So we are eliminating that. Only bacteria with the plasmid will survive. From that we can do check which of the plasmids will have the gene of interest. Then we'll finally send it for sequencing. Otherwise, if like the plasmids are commercial plasmids are made like that, so that the antibiotic resistance gene is kept separate from the MCS. If you are disrupting your antibiotic resistance gene only, then it will not grow. The role of the selectable marker is that it helps the bacteria grow on an agar ampicillin plate. If you take away its purpose, then there is no role. That is how it is. Okay. So if they will conclude the session here, if you do not have any further questions, we can conclude the session over here and we can meet again next week, same time. Okay. So we can stop the session. Thank you.
Thank you,
Thank
ma'am.
you.